And you can see here, this is the frequency spectrum of electromagnetic radiation, and you can see that there are different parts of the electromagnetic radiation that the imaging modalities apply. So on one side, we have the ionizing modalities like X-ray, nuclear medicine. On the other, we have the non-ionizing like ultrasound, MRI, and the optical imaging. And here, there is the I tried to make a kind of comparison or to give you most of the properties of the in vivo imaging modalities that we have at the moment. So we can see, first of all, that there is different type of energy. For the PET, we look at the annihilation photons, 511 keV. For the SPECT, gamma rays, we look at gamma rays, CT X-rays, etc. We have different spatial resolution. And this gives us, what we can see, what details we can see on the image. Some of them, they are molecular, some of them are not molecular. How easy they are to translate them to the clinical situation, how deep we can go into the tissue. For example, the optical imaging, they are the ones that at the moment we use them only at preclinical stage and not in clinical, because we cannot go very far in a high depth in the tissue. And also they have a totally different molecular sensitivity. And again, a little bit more information here where you can see that some of them, they are very sensitive. Some of them, they are quantitative as they are at the moment, even though we have to do more to go into completely quantitative imaging. And some others, they are limited in molecular information and so on. Again, here you can see the relative sensitivity of imaging technologies. So we have the different imaging techniques here. You can see what we can study with these techniques, but also the sensitivity of these techniques are different for the different imaging modalities. That's why we cannot use X-ray, for example, to look at signal transduction or stem cell function, as it is at the moment, maybe in the future. Okay, so we are moving from the anatomical imaging, that's how the imaging started, to the physiological imaging, and more recently to what we call targeted molecular imaging. And as I said to you earlier, what we would like to do is to move from the population medicine to individual medicine. And this is the challenge here and is a new paradigm in cancer diagnostics. So what we would like to do is to find the right molecular probe, target specific contrast agent, or targeted drug, whatever you want to call it, for the right target to monitor the right disease in the right patient. So moving from generic downstream indicators to more specific targeted indicators, and moving from the symptoms to more biological process. And in order to do that, in particular in oncology, we are combine imaging modalities, and we do that because, as I said, we cannot, there are a lot of information, but each modality gives us different information, and taking the information from one modality is not good enough. It's not enough for a clinician nowadays to make a decision. That's why we are trying to combine modalities, and we do that either on a hardware, like we have systems. It's not easy to combine them. It took us a lot of time to do that, or we do it after imaging by getting the images and doing image registration and fusion, and then looking at the results after the fusion of the images. And in this way, we can get quantitative, anatomical, molecular, and functional information. But also what we do, apart for the multimodality imaging, is that we are trying to use predictive mathematical models where we provide patient-specific data, and then we try to validate these mathematical models of tumor growth and treatment response into the actual clinical using in the patients. And in this way, we are trying to combine the mathematical modeling and the information from the images in order to move forward into a new era of medicine. The imaging functions over a wide range of targets and time and size scales because of the biological and pathological processes that they are involved. The targets can be a physiological state, can be a receptor, can be an enzyme, can be DNA or mRNA. And the time scales can go from milliseconds, like for protein binding or for chemical reactions, to years, as it is for diseases like cancers. And the size scales going from molecular to cellular to organ to whole organism. So in some way, we need to find a way to push the limits of the technological developments of our detectors, of our scanners, so they can match the characteristics, the properties of the physiological processes if we would like to move forward into quantitative and precision medicine. And here I just use that as an example. This is actually the 7 hallmarks of cancer and their links to tumor metabolism. So in other ways, this shows how we transit from the normal cell to a tumor. So these are all the 7 hallmarks. Don't worry, don't try to understand. I'm just trying to say that the functional imaging techniques can actually describe these metabolic processes and the hallmarks at tumor level in the regions around the tumor, but also in whole organism level. And you can see here, for example, if we want to look at the metabolism and hypoxia, we use specific imaging modalities. If we go to proliferation, we use different modalities, and so on. So we need the molecular imaging techniques if we would like to look at the metabolic processes. Now, just to go a little bit after that in what my team does is that we are trying to develop emerging imaging techniques through innovative hardware development. And in this way, we generate a unique data at preclinical and clinical level. And these are some of the detectors, actually, semiconductor detectors that we developed in my team. This is a silicon-based detector. This is an amorphous silicon-based detectors. We also did an amorphous selenium, and this is our last baby, the cadmium zinc telluride detector. And if you look at this graph, you can see that the detection efficiency with respect, it's the detection efficiency to the X-ray and to the photon energy, yes, to the X-ray energy. And we use different semiconductor materials, as you can see here. All they have the same thickness of the material, so we are looking at the same thing regarding the detection efficiency. And you can see that if we use silicon, we cannot go very far. We cannot go far from the 60 kV, 70 kVs. But in medical imaging, we are talking about 140 kV for CT. We are talking about 300, 511 kV, 662 kV, and more. So the silicon is not good enough for looking at for using it at modalities, at medical imaging modalities. But all the other semiconductor detectors, you can see that they are having better and better detection efficiency. So what we are trying to do is we are trying to look at these different materials and to see if they can be transferred in detectors, and from the detectors into the scanning, into a scanner. So we will be able to do some new imaging techniques that they are going to give us some new diagnostic information. So what we do is that we have a dynamic, as we call it, hardware development approach. It is unique in my team, and we are trying to do theoretical and experimental investigation. So we start with a theoretical investigation where we are starting all the way from the detector. We develop the detector. We look at the design specifications, pixel, thickness, everything in the detector, how the electrical field, the electrical field behaves within the detector, how what is happening when the radiation goes through the detector, what kind of signal we get out of the detector, all this, and then we are going all the way to the system geometry. So after we are quite happy with the detector, we'll put it into a system geometry, we build the geometry of the scanner, and then we start and in this way, we are going very close to the real clinical conditions, and then we can look at the images. So we do image reconstruction and several other things, but also we are looking at the dose at the same time. So we are, through our Monte Carlo simulations here, we can look at the dose, and we are ready after that. So when we reach the point that we say that's how the scanner would be if we would like to move in a new imaging technique, we are very close to the reality. And then we are talking to the, excuse me, to the healthcare manufacturers. So in this modeling, as I said, we have everything from the detector, where we look at the geometry, how we are going to develop the geometry, how we are going to do the pixels, the thickness, everything, but also we are looking at the charge generation, at the charge transport, because we have different processes there. We have drift, we have diffusion, charge sharing later. So we need to look at all this and see the signal induction by the end, what kind of signal we get out of it. So we measure what we call charge induction efficiency, which is actually the signal that we are going to detect. And also we do, as I said, the system. So we build the geometry, but also we build readout electronics, and we do image and dose assessment. And if it is an X-ray system, we also model the X-ray tube, or if it is a gamma ray imaging, then we use the radioisotopes that we have to use. And here is, as I said, we combine finite element analysis for the detector is a semiconductor. So we look at the semiconductor physics. We use the COMSOL multiphysics code. Then we are moving to the Monte Carlo simulation, where we use the gate and the Jean4, where we look at the, as I said, into the whole system, but also in the interactions and everything that happens there. We use LabVIEW and SPICE for our readout electronics, but also we either develop our own 3D computational phantoms, like the Debra phantom here, the breast phantom, or we use some other publicly available anthropomorphic or mouse phantom to, excuse me, in order to do our imaging and dosimetric assessment. And we do that because, of course, as you understand, in order to develop a detector, taking, for example, the cadmium zinc telluride material and having a pixelated detector by the end that can go in a scanner, it's a very expensive exercise. So we cannot just play with it without in the lab without making our theoretical investigations first. So of course we are going into the lab. Of course we have different detector geometries and from the detector manufacturers that we work very close with them. And then of course we validate what we find or we changed our simulation, but this is actually this accelerates the whole procedure. And the same with the scanner. Imagine we cannot build a scanner to find out that this wasn't the right scanner to do. So that's why we do this simulation. And of course, all our simulation now is connected, and it is a huge multi-environment software platform. And as you can understand, we use high-performance computing to do that. Okay, now, as I said, our very recent baby is the cadmium zinc telluride detector. And we do that because we would like to do something that is going to be a paradigm shifter in X-ray imaging. In X-ray imaging is the mostly wided medical imaging modality. However, we have challenges there. We have problems there. We cannot do soft tissue differentiation, something that MRI can do, but MRI is very expensive. Then we have problems between the primary scatter radiation and the contrast. Because we don't have the soft tissue differentiation, our contrast resolution is not as good as. And we have, in order to achieve the image quality that we want, we are increase the radiation dose. Or one or the other way, this is not what we would like to do. So we would like to move from the conventional X-ray imaging to what we call photon counting multispectral X-ray imaging. And it's going to be the next thing, the revolution in X-ray imaging. And we are moving from the anatomy and morphology, which is the only thing that X-ray imaging can do now with a very little of molecular information, to getting the more molecular information and quantitative information. And here you can see, for example, this is the first scanner that Jeffrey Hounsfield, the father of the CT, developed. And you can see here the evolution of the CT. So we moved just by upgrading the CT all the time, thinking image quality versus speed versus dose. However, now we reach the upper limit. We cannot go any further. So we are trying to move to something different. And this is something that it is well known from 1975. That was the paper by Alvarez and Makovsky, when they put the theoretical basis of what we call dual energy CT or multi-energy CT. So now we are on the first part, because we didn't have the detectors, but now we have the detectors. Now we do have the dual energy CT scanners into the into our clinic. However, the dual energy CT, by using two energies there, by looking at two energies, we can get information about two different materials. And we use different configurations. However, we still have overlapping of regions, overlapping of materials, and increase dose. So we are trying to move for this dual energy to multispectral imaging, where we are going to have one X-ray source, one detector, and then we are getting a tissue-specific quantitative imaging technique. And you all know that the the X-ray beam is a polychromatic X-ray beam, the beam that comes out of the source. It's an incident polychromatic X-ray beam onto the human being, and continues to be the transmitted one continues to be a polychromatic X-ray beam. But at the moment, we don't look at all the different areas of the spectrum. And as we know, the attenuation depends on the photon energy. And this energy dependence is different for different materials, tissues, elements. So the energy of each photon can give us additional valuable information for diagnosis and treatment. But in order to do that, we need to have a detector that have very sophisticated spectroscopic features. They have a very high energy resolution, so we can do this separation of the spectrum. And then we can move to tissue-specific imaging. And here you can see, for example, the elemental composition of the different, the different organs here, or tissues, or the different, or of blood, of the bone, of the brain. And if you look at here, where we have the attenuation coefficient as a function of energy, you can see that the difference between the attenuation coefficient for different material is largest at low energies here. So we need to find a way to get this information. And we do that with what I said, a photon counting detector, where we count every single photon, and we get all the information that this single photon contains. And this is a semiconductor detector, the cadmium zinc telluride detector, that we use in order to get the signal out of it. You know, the X-ray is going through the detector. And then we have different readout electronics now, where we have different thresholds, so we can separate different bins, different energy bins. We can separate the spectrum, and also we have counters, and we count, collect the photons of each different bin. Okay, I don't need to. However, it is not an easy thing. We need to do a lot of things in order to be able to do that, to move it into the clinic, and that's where we are working. We have been working on, but we think that this really is going to bring a revolution in X-ray imaging. As I said, there are several technical problems, and these are mainly comes from the fact that all the X-ray imaging modalities, they use very high photon fluxes. We are talking about 10 to the 9 photons per millimeter square per second for the CT, and the mammography 10 to the 6, 10 to the 7 photons per millimeter square per second. But because we need to have very small pixel sizes of our detectors, because we need to have high spectral resolution, this means that a lot of problems are created, and this is where we have been trying to solve. But I would like to show you just what we can do actually with a multispectral imaging, which we cannot do with X-ray imaging as it is at the moment. We can simultaneously, in one scan, we can look at all these different agents, and also we can look at different tissues, like a soft tissue, a bone, a blood, whatever. And this actually, as I said, makes it more quantitative, give us more information, and is a tissue-specific. It's a patient-specific imaging. And here you can see how the contrast changes. This has breast samples that we did in my lab with the first generation of the detector that we had, that we developed. And you can see these are for different energy bins, the same part of the breast tissue, and you can see how the contrast changes. And this is what we are trying to do. We are trying to catch the small difference in absorption. And this is a task preserved by MRI. But if we manage to do it, I think that we are going to be the winners over the MRI. And as a final thing, I would like to show you this very complicated readout electronics, which has been developed at CERN. We are part of the MEDIPICS collaboration. And this is how a photon counting readout electronics look like in order to be able to move to this multispectral photon counting X-ray imaging. I'm not going to go into details on that, but you can see how complicated it is and a lot of things that we have to do in order to be able finally to move into, to translate it into the clinical situation. Okay, so I think that I'm close to the only thing that I would like to add here is that, as we said, there is the imaging is multi-level and give us different information for different things. But so we have information for all from different parts, from different at different levels and scales. But also at the same time, we have information from genes, from metabolics, from proteomics. So we need to find a way to put all this together so we can move to precision medicine. This is not an easy task. Again, this is something that we have been working on, and we hope that sooner or later we will be able to do it, and then we are moving to precision medicine, where we can do so we're moving towards a model of holistic, cost-effective, personalized, and proactive patient care, and we are talking about accurate early detection, but also precise and targeted therapy. Okay. Just to say that at the moment, as the modalities are, as I said to you before, we get images, but we get high noise, and this, of course, limits the spatial resolution. And this doesn't give us reliable quantification of the quantitative metrics that we put in every image. So the challenge for us, the detector physicists, is to build imaging devices that are also measuring devices. And that's what we do. An example is that what I said to you with the photon counting multispectral X-ray imaging. We build a detector and readout electronics that is going to give us very quantitative diagnostic information. And in this way, we are moving from symptom-based to patient-specific diagnostics. So we are moving from a one-size-fits-all approach to precision medicine and personalized care, but this is something that it cannot be done only by biologists or by medics or by physicists or by chemists or by engineers. I started with an elephant. I finished with an elephant, just to show you that in order to achieve what we would like to achieve, it's going to be a joint effort between all the different disciplines. And this is what we do in Institute of Cancer Research and Royal Marsden Hospital. We work all together in order to move forward. And just to remember my Greek origin, I can tell you that Hippocrates knew about precision medicine long before anybody else. It's far more important to know what person the disease has than what disease the person has. Thank you for your attention.
Thank you very much, Dimitra, for this very informative talk. I hope our students enjoyed it. And I think now the floor is open to questions.
I have a question.
Yep. Hi, Tom.
By any chance, could I get your email so I can ask you any questions if you have any free time?
Sure. It's dimitra.darambara@icr.ac.uk.
And please, could you type it?
It's okay. Let me go back into my. It's in the first. I think that I have it on the first slide. If I manage to go there, yeah, it's here. Yeah.
Okay, thank you very much.
No, my pleasure. Yep. Anyone that would like to ask questions or is interested in doing research, here we are. Yeah, please contact me. It's my pleasure.
Thanks, Dimitra. Anybody else? Any comments?
I have a question.
Yeah.
How much success have you seen with the hardware development approach so far?
Sorry, what?
You said your team were working.
Yes.
On the hardware development approach, and that it was
yes.
team. So, how much success have you seen with it?
Oh, we are by then we are successful. Whatever we designed, they are scanners now. The silicon pad detector, which is this one actually that we designed, it's on a low angle X-ray scattering used for biology. Then the large area detectors that actually we were the first starting working in the UK and develop it with the manufacturers, is in the scanners now. In all the X-ray imaging scanners. That's the large area morphosilicon detectors. We also used in morphoselenium detectors for digital pressed tomosynthesis, as they are there. And now we have been working on the cadmium zinc telluride, which is going to take us longer than the others, but I'm quite confident that by the end we are going to be able. I have to say that we don't work alone. We are doing all the physics and all the simulation and all the experimental validation and investigations in the lab. However, we work very closely first with the detector companies, so we can get information about the materials, and also we ask them to design several devices for us so we can test and see how close to our simulations are, and then change accordingly. Also, when we reach the point of a scanner and we start working on a benchtop scanner, then we work closely with the healthcare manufacturers.
Thank you.
Thanks, Georgia.
I have a question as well, actually, now thinking about this. With the X-ray imaging, is it correlated to the current status of what's going on with the CERN team, where they've been able to show different X-rays with colors now?
If the X-ray imaging is related to the CERN team, you said?
Yeah, because I know the CERN team have created X-ray images that can also display colors now.
Yeah, you're talking about the Medipix.
I think it was 4 or 5 months ago.
Yeah. Yes. We have been working with the Medipix collaboration, which is they are working on the readout electronics. Yes, on the photon counting detector. That's complicated circuit that I showed you. And yes, there is a part of one of the members of the collaboration is Mars. Mars is a company in New Zealand, and they created the first small scanner, and they had their first human images out, but they are only for bone imaging, while we are looking mostly on the cancer imaging, where that's the most difficult situation is.
Okay, thank you for that.
I mean, have you seen this the hand image of from Mars? Is this what you were referring to?
Yes, but there was also an image that they had where they were showing what appears. No, actually, yeah, it was the hand. It
Yeah,
shows
the hand. Yeah,
it's got like
yeah.
different colors attached to it.
Yeah, yes, yeah. That's the small scanner that they created for, and we work very close to Mars as well. And hopefully, we hope that we are going to have one of their systems into our lab, one of these small scanners into our lab, and we will develop it further for cancer imaging application and for imaging applied to radiotherapy as well. Because, of course, imaging plays a great role in radiotherapy for planning, for response to the therapy, for all the different, for the dose that we give, for all these things that we do in radiotherapy, in the modern radiotherapy.
Thank you for that. It's really interesting.
Yeah, it is interesting. And if you are interested in research, talk to me.
Any other questions, guys? I actually have a question, Dimitra. You mentioned personalized tumor characterization and prognosis, and I was wondering whether artificial intelligence would play a role in this regard.
Yes, I'm sure that it's going to be one of these. It's going to be part of this big picture where we are going to have all the different data, and also. Yes, we believe that artificial intelligence also is going to be part of it. And we actually work on this thing by trying to train a system using some data of imaging for radiotherapy to see what we can get out of it. But yes, I'm sure that artificial intelligence is going to play a role in that, big circle where you have everything and you have to combine everything in order to move to personalized medicine. Yeah.
Yeah, because there's so much data that a human being can analyze. But if you have artificial intelligence,
sure.
then
Sure.
the range of data you can analyze is much wider.
Yeah, that's one of the, yes, I think that's this is going to be important. Yeah, definitely.
Sounds good. Any other questions in terms of the scientific part?
Anyway,
not,
anytime that they like to ask questions, they can send me an email. There's no problem.
I have a question.
Oh.
With the ability of the body bones in terms of X-ray being able to absorb or basically send the image, back so we can see it. Is it possible that with the higher photon count, we can actually understand, based on the absorption rate, the particular materials inside the body without having to, take the parts of the body out?
Yeah, absolutely. This is the whole thing behind the photon counting multispectral X-ray imaging, and this is what we would like to do. We would like to look, as I showed to you, there is you can see that there are different different elemental composition per tissue, per bone, per blood, per whatever. And this is what we would like, because the photon counting multispectral X-ray imaging is going to be at atomic level. That's where we are trying to get this kind of information. We are going to try and do different material decomposition. Yes, that's part of the big thing for multispectral.
Okay.
Yeah.
Thank you for that.
This is going to be one of the two big things. The elemental composition is going to be extremely interesting. And also we would like to look also at the different elemental composition and ratios between elements and all these things between tumors. Between tumors, different stages of tumors, of a tumor, and so on. So yeah.
Okay.
but it's not easier to do the material decomposition, and that's the more on the imaging reconstruction guys, the software guys, but we work close with them as well. And yeah, it is. I think that's the beauty of one of the beauties of the multispectral imaging. Yeah.
Thank you, Dimitra. Could you please, guys, allow me to move towards careers questions? Dimitra is an expert in medical imaging, so if any of you would have any questions about how could one enter into the field, please fire away.
By any chance, can someone be a lab rat?
Sorry?
By any chance, can someone be a lab rat as a part of a job in it?
Sorry, I didn't hear you very well. Sorry there.
I was just wondering, like, if you're going into the career path and you wanted to test the machinery, could you enter the job in a way as kind of like a lab rat to be the person who's being tested to see how it displays?
I'm not quite sure that I understand your question now.
Like,
maybe
say, like, in terms of like, if we do it to the normal sensible sense of when we're playing games.
Yes.
And there's like a beta test where a select group of people get to be the testers of, say, the game coming out.
Okay.
So
okay.
like.
Okay. Now, okay, that's what I wasn't sure that. Okay. As all the medical devices, okay, there are a lot of guidelines, if you want, that we have to follow. So when we reach the point that it is ready from the benchtop imaging, the imaging scanner, the small scale scanner that we have, to move into a large scale scanner, then the medical manufacturers come in. They take it, they do it, and then, of course, they come back to us. To us, I mean the Royal Marsden Hospital or any other hospital, to do what we call clinical trials. And the clinical trials, they are in different stages again. So, yes, then we have human beings that they, you know, with their consent, of course, they try. They can do the scanning.
Okay.
If this is what you meant, I don't know.
Yeah, the multilinear stage is a trial.
Yes, yeah, we are going to the trial, and the trials, they are in different stages. Okay, now let me tell you something. That's a secret, okay? I'm joking. When we really do when we're reaching the point that we are going to have a scanner, usually we by ourselves try sometimes the scanner before before it goes to the patients. But of course, it depends on what kind of procedure you are going to do. It's a radiation imaging here. We are talking about doses. So we have the other good thing with the photon counting, because it is photon counting, we are going 30 to 40% down in dose without affecting the image quality. That's another beauty of the photon counting multispectral X-ray imaging. So, but in order to get a scanner, in order to get or a drug, if you want, a drug for cancer, in order to get the final approval by the government, by the regulatory bodies, then we have to go through clinical trials with real patients and with the consent of the patients.
Okay. I just find it interesting because we're quite fair. It's probably going to take is it normally like a few years or so before those trials go through?
Oh, yes. As I said,
I was kind of looking forward to getting my own X-ray.
moving from the bench top all the way to the scanner that goes to that is ready to go to the clinical situation, it's a very long procedure, and it's not only the procedure at the time that you spend to develop it and finally reach the point of having a scanner. It's also the fact that it has to go through clinical trials, and it starts with a small number, a small cohort, patient cohort, but then we have to go to large numbers. In particular, because, for example, if there is something for screening, like mammography, and we do something different with, for example, distal breast tomosynthesis, we have to go at the population level because it's a screening, yes? So we need to prove that it can go to clinic. So, as I said, there are different clinical stages, but it's a very long procedure. By the time that we start moving from physics to start doing the technological development, all the way to be ready to the clinic, to be used in a clinical environment, it's 15, 20 years. It depends on the application. It depends on the scanner.
I've got a question if you're going to implement AI into this.
Mm-hmm.
If you want to implement AI into this, wouldn't you need servers, or are you using AI that only has particular features that it's supposed to display? Thus, you can neutralize the idea of using the servers.
Yeah, I mean, okay. I'm not the software person, but yes, I mean, we, at least with the AI, we work with Imperial College, with the guys there that they do the AI. And yes, they use servers, they use their own things anyway. They use high performance computing, if this is what you're...
Yes.
Yeah.
But Dimitra, can I just ask you, is the Institute of Cancer Research offering work placements to students?
Yes and no. I mean, it depends. You're talking about undergraduate students, yes?
Undergraduates, yes.
Yeah. I mean, there are areas that, yes, we can offer, and there are areas that we cannot offer. Mm-hmm. But it's not something that we are against it. We like students to come and spend time with us. Just we have to be careful to which, where they can be, where they can go, because of course, some of the areas, they are very, they are protected. Mm-hmm. And you can imagine, I mean, for me, all these things that I do, I sign non-disclosure agreements with the companies. So in order for a student to come and work with me, I have to get their approval. Mm-hmm. Or I can. It depends on which part of the research, but it's the same more or less. But generally speaking, we are not against. We are happy to.
So what would be the official process? Would they send an email to the institute directly, or would they send an email to you asking for a placement?
They can do both. I mean, okay, if they are physicists, it's much better to send it. Okay, they can approach whoever of us in the physics department, depending on the area, of course. They want magnetic resonance, nuclear medicine, X-rays, multimodality that I do. All the different areas that we have there, and they can directly approach by email the team leader, mm-hmm. or they can send an email to the physics secretaries, where they are in charge of then they can see, okay, this person is interested in MRI. Then they can send it to us, and then of course we all together try and find out how how we will do it with ICR being happy as well, and Royal Marsden, yes, because we have Royal Marsden there as well, and we are the only joint department between Royal Marsden and ICR, because physicists work on both, either as clinical scientists or as research scientists like myself.
Mm-hmm. Okay, sounds good. Thank you.
Yeah.
Any other advice you may have for physics graduates on how to enter the field of medical imaging?
Okay. First of all, what I will say is doesn't matter which part you are going to do. To become a physicist, you have to have a passion with physicists. Otherwise, you don't do physics. You do something else.
Sure.
So if you have a passion for physics, you will do it. And okay, there are different ways of doing medical physics. You can follow the academic or industrial research path, where you don't you actually, if you have a good first degree, you can see what the universities get out of as projects for PhDs, and you can apply, and one can apply and then follow that part, doing a PhD and a postdoc later if this is what they want, or moving to industry to work in industry. Mm-hmm. The other part is to be clinical scientist. But this means that 80% of their time is going to be in the clinic, where they have to deal with all the things there that they include physics, like look at the at the different scanners, doing the quality assessment, helping with the doses, doing the planning for radiotherapy. All the different things there. Also, they do computing as well. They do programming and analyzing data. And this is something that if you would like to be a clinical scientist, you have to follow this STP program, which is a 3-year program, and there are some entry requirements regarding I think that you have to have either first or 2.1, but I'm not quite sure because that's not my my part exactly. And then there is there are 3 training universities, that there are 3 training centers, at least in there are 3 or more. I think there are 3. It's the King's College, is the Liverpool, and what is the other one? And there is another one anyway. Glasgow. Mm-hmm. Oh dear, there is one in Scotland, which I can't remember now. Anyway, and you have to apply. There are usually the application starts in January. It is quite competitive. There are a lot of people that they apply, so you have to go through interview and some kind of assessment, and then you can go and do this 3-year training, where you have to spend time at the university, where you do your MSc in a way. You get an MSc as well on medical physics, so you have the taught courses, so you cover everything on medical imaging and on radiotherapy. Mm-hmm. And also you have to get some other skills, all these things that they have in a program. And also they do either small projects or by the end a large project, a research project. And after that, they are ready to be hired by, to apply for a job in a hospital and work as clinical medical physics scientist.
I see. What does STP stands for?
STP is the Scientist Training Program.
Oh, I see. Okay.
Which is an NHS scientist training program, and it's for all the clinical scientists. And then they have, of course, the part that it is on medical physics.
I see. Okay, thank you.
And the other thing, of course, that you can also go to the clinical part is to be a clinical scientist. Is you can do a PhD that it is very clinical oriented, and then you can do another 3 years within a hospital doing working on projects that they are you know, then you're in a way you will be trained on the clinical part, and then you can be a clinical scientist.
Mm-hmm.
The only thing is that the one with PhD is a longer, of course, path, because you have the time that you spend, and then you have to do another 3 years in creating your portfolio. You know, doing a little bit of diagnosis, a little radiology, radiotherapy, nuclear medicine, different parts, and then you will be able to get then you can work as a clinical scientist.
I see. Very interesting. Thank you.
Yeah.
Guys, any other question in terms of careers? No?
Still, if they would like to ask something, I mean, they can ask at any time. Yeah. And for the clinical scientist part, even though it is not my, I can ask our colleagues that they are clinical scientists, and also when you do this STP training program, you are employed as a fixed term, under a fixed term contract, and you paid a salary during the training.
Mm-hmm. Oh, that's good to know.
I actually have a question now.
It's interesting, yeah?
So I have applied for the STP this year, and I've been shortlisted twice. But is it possible to get work experience in the clinical sciences department?
When?
Maybe summer.
Okay, that's something that I cannot answer because I'm not a clinical scientist, but if you want, send me an email, and then I will forward it, or I will ask the head of the clinical scientists, you know, the clinical physics guys, which is next door to me. She's her office is next door to me, so I will ask her, and I will let you know, because that's something I know that from time to time they have, but I don't know the details on that. But yes, I can ask them.
Thank you.
No problem.
Thank you, Dimitra.
That's okay.
Anything else, guys?
Anyway, at any time that they would like, they can do it. They can ask me.
Well,
if I don't know, I will find the person that knows.
Yeah, sure. Well, if there are no more questions, I would like to thank Dimitra for this wonderful presentation and informative discussion we had. And if everybody could open up their mics and give a round of applause.
Thank you, guys. Thank you. And thank you for the invitation. And also, I would like to wish all of you good luck with your exams. I know that you are in your final year, so good luck with your exams, and all the best for your career. Just follow your dreams.
Thank you, Dimitra. That was very nice.
Thank you.
Well, thanks very much, everybody. That was the last session of our physics seminar series, and we will reconvene next year. So thanks again very much for attending.
Okay. Bye.
Bye. Bye, everybody.
Bye.
Thank you.
Bye.
Bye. Okay. Go out.
Thank you very much, Dimitra.
My pleasure. No problem. Anytime.
That was very nice, actually, because I think you received quite a few questions. We had almost 20 minutes discussion. Yeah,
that's great. And as I said, I'm happy to answer questions. And if they are research, okay, I can deal with them. But if there is anything on a career, on a clinical scientist, again, we do have them in Royal Marsden. So we are one department, so I will find out.
Well, thanks very much again.
Okay, my pleasure. Okay, bye.
Bye, thank you.
Bye.